Okay, I think recording is already in progress. Is that correct?
Yep, that's correct.
Okay.
I just started.
All right. Yeah, sorry. All right. So welcome everyone to our VICC VMTB series. It's my great pleasure to introduce Till Hartmann. He's a computer scientist by training and did his PhD in Essen and Dortmund on bioinformatics, and now joined the core unit bioinformatics here in Berlin. He's been working on variant annotation and consequence prediction for rare diseases and oncology as well. And I'm very happy that he's here to present Mehari, his recent tool. So go ahead, Till. Stage is all yours.
Yeah, thank you very much for the introduction. You already told everybody who I am. Okay, so I'm Till. I will talk about Mehari, which is basically a tool very similar to Ensemble's Variant Effect Predictor, VEP. And yeah, I will jump right in. So first, I want to make a little distinction between variant annotation and consequence prediction, because at least I make a distinction between them. So to get this cleared up from the start, for me, variant annotation is taking a variant, its location on the genome, its chromosome position, the reference and alternative allele, and basically doing a database lookup and just annotating whatever the database says. So in this example, just, okay, we have this fake chromosome, position 7, reference A, alternative C, and nomad frequency will tell us, okay, this we've seen 50 times or something. And this is a very simple lookup. That is easy. Now, for consequence prediction, we take the same information, but now we have to somehow match that to, let's say, a transcript model and combine these two things and integrate the actual change from the reference to the alternative, and then have a look at what that changes in the transcript sequence and what ultimately what that change is in the protein sequence, if we translate that. And this is what we're mostly concerned with here. So, as I already mentioned, there are, of course, a lot of existing tools in this landscape. So we have VEP, which is very widely used. I put community standard here, but I mean, it's a little bit subjective, of course. It has been around for a long time, very feature-rich, has a lot of plugins which are used. There are a couple of other tools, so SnpF, ANOVA, JANOVA, Nirvana. They fill slightly different niches, but do mostly the same thing. Some of them are only for RefSeq databases, some are for Ensembl. Some of them have custom output formats and so on and so forth. Now we did, well, yet another tool, which is Mehari, which concentrates on doing like an HGVS first approach. So HGVS notation, we first normalize and then we do all of our calculations based on the HGVS notations. Why are we doing this? We do this because we want to eliminate any ambiguity between the representation of the variant and the predicted effect. You will see later on what I mean with this in more detail. Also, we wanted to make it quite fast. So VEP is notably rather slow, and we thought, okay, we can do probably better because we have large-scale data sets and the whole genome sequencing, and then you have thousands of samples, and you don't want to wait for weeks. I mean, if you have the time and the resources, sure, but you can do better and do it in a couple of hours or days. What we also get from this is that we can easily handle selenoproteins where you have an additional, the selenocysteine, which can sometimes be interpreted as an additional stop or, well, not as a stop, depending on actual context. And also VICC gapped exon alignments, which sometimes occur and sometimes don't. Okay, let's talk about ambiguity. Take this variant, for example. So this is on GRCH 38, chromosome 3 at some position, and this is basically a deletion of 5 bases. If you put that into your VCF notation and do a VCF normalization, so variant call format, I assume if anyone doesn't know something, please ask, because I make some assumption on what you know, and yeah. So in VCF notation, normalizing this gives you the first line, because VCF normalizes to the 5' end as far as possible. HGVS has the 3' rule. So in HGVS notation, everything is shifted as far as possible to the 3' end. And that already leads to problems when talking about variants. Like if one party talks about the VCF variant and the other party talks about the HGVS normalized variant, you may be confused. Yeah, so this is just from the HGVS website stating the pre-prime rule. So for any description at all, so genome, gene, transcript, protein, you always assign the most 3' position to have been changed. Now, if we take this genomic sequence and pick a transcript that corresponds to this genomic location, this makes it even more complicated, especially if the transcript is a reverse transcript, because then what the 3' rule tells us is, okay, we need to push it as far 3' as we can, which in this case means from -13 to -9. So this is before, this is intronic sequence. And yeah, VEP will not do that. But well, this sometimes is a matter of choice and preference, but we try to stay as close to HGVS as we can. And if that's the recommendation, we do it and base all our predictions on that. Exactly. So VEP can also do that. The thing is, you have to be very careful with your CLI options. So here's a little overview. I put this same, this you don't have to read this. I put this same variant into VEP with different options, and you get a different combination of consequences of HGVS notations and so on and so forth. Technically, a lot of these aren't allowed because the documentation says, okay, shift HGVS can only be enabled if the other two that I mentioned here, shift 3' and shift genomic, are disabled, but it still lets you do it and something different happens. So be aware of that. Okay, so yeah, HGVS first. Then I wanted to talk about performance. So for this, I picked a little benchmark. And what we see here is that we are about 40 times faster than VEP on this specific benchmark, which I can tell you about later. Sometimes it's 80%, 80 times faster, depends on the exact number of annotations you actually annotate and how much I/O overhead you have, and if you're doing Ensembl and VICC annotation and so on. But in general, you can go from an hour to a minute, very roughly speaking, which is nice. So now we can do very large cohorts in a reasonable amount of time. I also thought about just annotating every possible single nucleotide variant that you can have in a human genome, so about 9 billion or so, because that should be doable in a week or so. Might be interesting to do. All right, let's talk about consequences. I'm sure most of you have at some point perhaps encountered this little infographic from the Ensembl VEP website, where you can see in which regions have which consequences. This is a rough overview, and it's nice to read. But I was always confused what exactly that means. So I went in, and this is not particularly beautiful right now, but this is a work in progress. I annotated every position quite precisely. So these are the things where you can actually see I've taken the definition of the consequence term from sequence ontology and mapped that to this graphic. So this is a little bit more precise, I hope. What I will point out here is that VEP, or rather the other way around, Mehari is a little bit more specific. So VEP will only say 5' UTR variant, for example, while Mehari will differentiate between if this is a 5' UTR variant within the exon or the intronic part of that. Yeah, Manuela.
Can I ask a quick question right here?
Yes.
Sorry. Can you explain the different high, moderate, low definitions there? Because there seem to be two.
Oh, these are the same, just rotated. So you have to do less cognitive mapping of turning your head, because sometimes they're turned.
Okay.
Yeah. Okay, sorry. Yeah, and
And
these impacts, the impacts are the same that VEP uses for its consequences. So each of these consequences has an associated impact, and this mimics that.
okay, yeah, thank you.
Yeah, so start lost is bad. So this is impact is high. Yeah.
Yeah, okay, thanks.
Yeah. What I will also point out here is that, for example, the splice donor fifth base variant, giving you a little bit time to locate that, because I don't think you can see my pointer. Is contained within the splice donor region. So sometimes terms contain other terms. And this leads me to the sequence ontology. So both Mehari and VEP, and roughly speaking, the other tools do this mostly as well, use sequence ontology terms for their effects. And these are actual things that are part of a graph. So they have relationships to other terms. In this example, I've highlighted the in-frame deletion. And this has two children, namely conservative in-frame deletion and disruptive in-frame deletion, which tells you, like, does the deletion happen within a codon or at the start of the codon, basically. And in-frame deletion itself is an in-frame indel, which is an in-frame variant, which is a protein alter variant, and so on and so forth. And well, in my time watching people filter VCF files and look for effect from consequence predictions from VEP, I've only ever seen them use hard-coded strings. And well, I'm a computer scientist by trade, so I thought, why do we not use the dependencies, the relationships that we have? So short detour, we can filter for that. So there's this tool called VMBrain, disclaimer, I wrote that. And that has support for filtering for sequence ontology terms in like annotations. So the way to do this is, okay, you say, I have this list of annotations in my annotation field. And if any of these annotation terms so missense variant and example or disruptive in-frame deletion, if any of them is a in-frame deletion in this sense, so disruptive in-frame deletion is a in-frame deletion, then I want to keep that. So yeah, that's just a very useful thing, because if I had only looked for does the annotation match the term in-frame deletion, I would not have found this, because this is a disruptive in-frame deletion. Yeah, just a little pointer on why I think sequence ontology is great, and we should use it more. Okay. I will quickly talk about validation in the sense that I wanted to look at, okay, what does Mehari do? What does VEP say? To that end, I took the ClinVar VCF files and annotated about 3 million ClinVar variants with both VEP and Mehari, only on the Ensembl transcripts and GRCH38, and only for main transcripts to make the comparison a little bit easier. And then I looked at what were the sequence ontology terms that we annotated, how do they differ, and made some statistics about this. Now, because I said like sometimes we have more specific terms like disruptive in-frame deletion instead of in-frame deletion. For the comparison, we look for the most specific terms. If there aren't, we make some special cases for, okay, VEP does not ever say disruptive in-frame deletion, only in-frame deletion. So we will say in-frame deletion and disruptive in-frame deletion is, for the sake of comparison, basically the same. You can have a look at the workflow itself, but let's talk about the statistics. So if we look at the sequence ontology terms that VEP said and Mehari said, we get quite high concordance depending on how you look at it. If we say, okay, any difference between the two sets of annotations is relevant, that's basically the symmetric difference, then we get to like 97.5%, 0.6% of concordance, like only about 70,000 variants were disagreeing in that sense. If you only look at what the impact of these annotations is, if they are different, then only about 3,000 variants remain. And if you're only interested in, okay, in the cases where VEP and Mehari agree on something, there are only 12 cases where VEP and Mehari don't ever agree. And I'll come to that. Yeah, so I was pretty pleased with that. Here's a couple of the differences that are interesting. I mean, it's a lot to go through 70,000 differences, so I've grouped them a little bit and tried finding categories that work. So sometimes it's just a difference in specificity. For example, Mehari will report coding transcript in-frame variant in addition to other terms, as well as splice donor region variant in addition to splice donor fifth base variant. We only have to do that because the sequence ontology is lacking there. So in theory, splice donor fifth base variant should be a child of splice donor region variant, but it isn't. So this is why Mehari also reports both of them. But already, like the additional information, this is a coding transcript in-frame variant, accounts for 51,000 of the 70,000 difference we see. And I don't consider that to be problematic. Differences in interpretation are a little bit more interesting. This boils down to sometimes comparing, like, if you look at the sequence ontology definition of the terms, they can be a bit vague. And yeah, VEP will sometimes say if we encounter a frameshift variant that this causes a stop gained or stop lost, while Mehari will say this is frameshift truncation or frameshift elongation. Because the frameshift causes the feature to either be shorter or longer. And stop gained and stop lost, if you're pedantic, are defined in terms of there is a change within the actual stop codon, or basically the change directly modifies the bases that make the stop codon. So that is a bit of a different interpretation of the terms. Then our support for selenoproteins causes differences because we say this is a rare amino acid variant or a selenocysteine loss, while VEP says stop gained, which is usually correct, but for some selenoproteins this is not correct. Then there are funny. I call it funny. It is not funny if you consider that people may use this for diagnostics, but VP sometimes reports start lost after the start codon, or stop lost or stop retained after the stop codon, which is technically not wrong if you have an additional methionine, so an additional pseudo start codon somewhere in the transcript. You can lose that, but it's not the start codon. Same for stop lost. If you lose a stop after the initial stop, it doesn't really matter. Then these 12 completely disagreement cases. These are all cases where VP says this is a frameshift, and Mehari says this is a splice donor variant. And if you look at the corresponding ClinVar entries, you will find that these likely are the splice donor variant. The rest of the cases are basically what I think are normalization errors, where VEP's behavior in how it handles ambiguous positions, especially in splice regions or intron-exon boundaries, are a bit off. But I'm willing to be proven wrong, or yeah, perhaps I'm using it incorrectly. It's totally possible. So sometimes you get intronic consequences in exonic regions and the other way around. And one of my favorites is when you get a splice donor fifth base variant at not the fifth base variant in the intron. So this is one of the cases where the reported HGVS notation does not agree with, I assume, what VEP's internal model is. So VEP will do something different than it reports at the end. And this does not happen with Mehari because we do the HGVS notation first. So we have no reason to call the 10th base a fifth base variant. Then there are like quite a lot of VEP not reporting UTR variants. I'm not sure why. I haven't figured that out. And the start lost and start retained at the same time cases. I think a couple, one or 1, 2, 5 in that order. Well, one of them is clearly right, but you have to figure out yourself if you see that case. All right. Yeah, that's about it about the validation. I quickly want to show you how to use it. So Mehari has both a command line interface as well as a built-in REST server with an open API specification. If you were to run it on the command line, here's how to do that. Say, okay, Mehari annotates sequence variants. It does have a structural variants version as well, but I'm only talking about the sequence variants here. Then you feed in your reference, a transcript database, which you can download from GitHub there's a README description on how to get that and how to build that if you need to. And then you can enable some options which mimic some of the options VEP has available. So only reporting the most severe consequence by either grouping by gene or by the allele or by transcript. This is not enabled by default. You can also restrict your transcript that are used for the annotation to specific things like only main select or the longest in this case. And if you restrict the transcripts you annotate, you can also say, I want to annotate all of the ones I pick or only the first that matches the order of transcripts I specified. And then you put in your VCF and get a VCF out or gzip VCF or BCF or whatever. Should be pretty straightforward. And then there's the REST API. So basically, you can just have a server running somewhere, multi-threaded using Actix Web, and then you can put in your genome release chromosome position reference alternative, and what you get is a JSON response, which looks somewhat like this, just as an example. And it will, because you have an open API specification, this is all very cleanly defined, and you can easily build applications for that if you want to. What we do not have yet is a plugin interface. I have written a draft, but VEP has a lot of plugins, and most of them are written in Perl. We do not have any plugins yet, but it should be very easy to write them because the plugin interface draft that we're currently implementing uses WebAssembly. So any language which supports or targets WebAssembly can be used. That includes Python and potentially R, I don't know. There are a lot of languages out there which support WebAssembly. That is the plan, but it's very much a draft at the moment. So if you need anything urgently, please let me know. We can make it happen. So just as a quick disclaimer, right now, if you do research and exploratory analysis and want performance or need to absolutely ensure that the HGVS notation that you hand out is in tandem with the actual effect prediction, and want a very lightweight, very fast tool, then Mehari is fine. But if you need plugins or regulatory regions annotated, or have like an established validated pipeline, then please do keep using VEP. And that's roughly about it. So quick summary. We have our Rust-based command line interface and REST application and REST server. For variant annotation and sequence ontology term prediction. I mentioned variant annotation here again because we do annotate, we have databases for Nomad and ClinVar VCVs, so we can annotate those, but the focus is on the term prediction. Why did we do it? We wanted better performance. We wanted to be able to do large cohort studies and quick re-annotation and on-the-fly annotations and stuff like that. And we also wanted to see, can we uncover any corner cases in VEP? And of course, the HGVS consistency again. How did we get here? We have pre-built transcript databases. We have a detailed consequence model. We use HGVS RS, which is a direct translation of the Biocommons HGVS. Yeah. And it's available on Bioconda. There are SnakeMake wrappers for both the seqvars annotation and downloading databases. If you have feedback, please please have feedback. You can do so on GitHub. Is most convenient for me, at least. Any bug reports, feature requests, or contributions are welcome. And yeah, these are the people I worked with on this project. Manuel has since gone into industry, Max as well. So I'm currently the only one left. I would also like to thank like Dave Lawrence and Chris Hart and the Biocommons HGVS teams, because I had a lot of questions, and I mean the Biocommons HGVS team did a lot of work on this HGVS part, and we only translated it to Rust. Yeah, with that, I would like to take questions if you have any.
Yeah, first of all, thanks for the presentation and overview too. Yeah, floor's open for questions. You can also write them in the chat if you want to. While you're thinking about your questions, I would start with a couple. First of all, you hinted at the beginning that you did the benchmarking on performance. Can you detail a bit about on that?
Yeah, okay. So for the benchmarking, we took a genome in a bottle NA, the default NA 12787 or sample VCF calls for whole genome sequencing, which has about 4 million variants or so, and ran that through VEP and Mehari and also ANOVA, JANOVA, and Nirvana. And well, this is the benchmarking setup, and then we had a look at how fast that really is. And Mehari took like 1 minute and 20 seconds, and VEP 40 something minutes.
Yeah, but Nirvana is nowadays built into the Illumina Dragon, so I guess the performance is then not easily comparable. Or did you use a Dragon?
Yeah, okay, so Nirvana has a release from 2022, but Nirvana also writes a different format, so you're comparing apples and oranges anyway. But Nirvana is a little bit slower than Mehari. The only thing that was slightly faster is JANOVA, but JANOVA also annotates way less things. So yeah.
Yeah. Okay. Yeah. Right. That's probably hard to compare then.
Yeah,
Then
but
maybe
it's
a
valid.
sec.
I mean, we did all the comparisons, but still the caveat is all the tools annotate different things, write different formats, and it's a bit difficult to compare fairly.
Yeah. Second question would be to us with respect to your pre-built databases for transcripts. So, how are you ensuring changes in annotation, like in transcript annotations there, or differences for different genome reference genomes? Are you preparing all the databases yourself? Can one do it later on?
Yeah, okay, so right now this is a very human genomics focused thing because of my predecessor. I would like to make that a little bit more well, dynamic. But right now we build databases for GRCH 37 and 38 for both Ensembl and RefSeq. They are built in a GitHub CI, so they're continuously built. So if there's an update, a version update for some of that, that is also built. Everything is versioned, so should be easy to follow changes. You can build your own. I mean, the GitHub CI tells you how to do it. Basically, it has a snake workflow for building this. If you want to make any changes, you're free to do that.
So I wanted to ask you about like opportunities for collaboration. I saw Dr. Hart's name on there. I know that he has been working with the GA4GH variant representation specification. Have you been working kind of closely with that group, or want to I don't know, connect with that group at all? Because I know that they're pretty integrated with the VICC team.
I've mainly talked to them when I had questions about specific HGVS
Okay.
nomenclature
So it
problem.
should be in line with kind of what they're doing. It's just it's more so, yeah, you have the HGVS nomenclature being populated, but I know that their representation is a little bit different too, since it's kind of annotating behind the scenes.
Mhm.
Yeah, cool. Thank
Yeah,
you.
okay, yeah, sure.
But yeah, if you can think of any reason that you would need to collaborate with that group, I could get you in touch with everybody. But it seems like you've already been in touch with a couple from that group. So I mean, just that was the first thing that crossed my mind was like, oh, are you in the know with everything happening in the VIC? But
Yeah, I'm not in the know of everything
okay,
that's happening, but yeah, I know these people exist.
they exist. Cool.
I guess an interesting question would be how to continue forward with the more structure variance fusions. I mean, the things you're also working on for the annotation, Beth.
Mhm.
Maybe that's something to follow up on. I did get that right, that this is not currently part of Mehari so much.
There are basic structural variant annotations, but only very basic.
Yeah, so it would be like at the sequence level, correct? Not like a 100 KB deletion or anything like that, or would that be part of your infrastructure?
I do not know. That is not I haven't written that part of the code.
Oh, okay.
So
So
yeah.
not yet, but perhaps in the future. So yeah, we should keep in touch when we're creating specifications surrounding fusions and copy number variants or more complex kind of co-occurring variants as
Yeah.
well.
Do you have any other questions from the audience?
I guess my other question is, have you had challenges with the HGVS nomenclature in general? Sometimes I feel like it's not super human readable when you get to kind of the more complex things, but it's manageable if you have like a single nucleotide variant or something at the sequence level. So I want to kind of gauge like what your experience has been using the HGVS nomenclature overall.
I I mean, I haven't encountered very, very complex things. But most of the time, I mean, it is quite, I mean, it is structured. And as a computer scientist, I like that it's
You
structured.
like
And
structure.
I like structure. So, and it makes it easy to see what's happening if you spend a little time in exploring the structure.
Hmm.
Um, but I can imagine that there are very weird cases which I would never be able to read. Uh, so,
Yeah, I mean, once a variant gets really complex, then you start to look for other options other than HGVS because
mhm.
it gets really lengthy and not super human readable. But like on the like computer science side, it makes more sense to stick with the standard just so you get some kind of correct annotation. But there's kind of a balance when you're talking about like communicating what you're seeing either in a paper or in a clinical report versus like getting all the annotation right on the back end.
Mhm.
So I mean it's something that's not solved by any means, but it's
Yeah.
something that I don't know, I like to kind of keep a pulse on.
Yeah, maybe one more question. I mean, you said you use genome in a bottle, presumably mostly for germline variants, or did you like really try to compare the somatic versus germline
Oh, this
annotation? Are
was
there any differences?
okay. Sorry. Yeah. This was just for the performance benchmark, so I didn't even look at the results. It's just okay. How fast can we do it? Done.
Okay, no, but even for the comparison between VEP and Mehari, did you look into whether this is something which is specific more in a germline case versus the somatic case? Where would it be relevant?
So yeah, that data set was the ClinVar ECF file. So anything that's in ClinVar, I'm not sure what that.
And they expanded to somatic as well, so I don't know.
Yeah. So.
But that's rather new. The somatic is rather new. But hopefully that'll be a useful data set for somatic very soon. But I mean, really, I think what I'm seeing is that you compared the results from the ClinVar data set with yours, and that is, even though it's germline, I think it does translate well to somatic because we still need to look at variants from a germline perspective and need to annotate them in a way that people understand. So I think it translates well. It's just cancer biology has layers upon layers of nuances. And so yeah, we always need a baseline, and HGVS is usually a good baseline. So I think the work you're doing is very relevant.
Yeah, glad to hear.
Yeah.
Hey, I don't see any raised hands as of now.
I guess, oh, did somebody speak up?
No, sorry, that's my background here.
Oh, okay. I was wondering, kind of to wrap us up, like what do you see as like the future of this product, and how like what are kind of the next steps that you're looking forward to developing?
Okay. Yeah, I mean, I would very much like to make use of this in large cohorts where I know, okay, previously, if I want to run VEP, it would take ages to get through all of that. And now I get the chance to actually do it quickly, but still get very similar results to VEP. So much for usage. Like for coding, I really want to get this plugin interface done so people can contribute their plugins that already exist for VEP or can translate existing VEP plugins to this. That would be great.
Yeah. Awesome.
So if you have a large cohort, I would say contact Till if you wanna get started with a fast tour.
That'd be great. Yeah, thank you so much for everything. This is awesome.
Thank you for having me and saying that this is awesome.
Great.
Yeah. So thanks for joining. Thanks for presenting. And if anyone has any questions, I think Till would be happily answer your questions. And the talk will be available via YouTube. And maybe we can also put the link to your GitHub in there so people can reach it more easily.
That'd be great.
Yeah.
Yeah, I can put that in the notes for the YouTube
That would
video.
be great. Yep. Thank you.
All right. Perfect. Then thanks everyone and have a great day or evening.
Thank you. Bye.
Thank you very much.